Genome evolution. Evolutionary theory predicts that organisms that are derived from a common ancestor, they will share genetic signatures. Organisms that shared an ancestor more recently, they will be more similar. So this is the concept of the evolutionary theory, that those organisms which are derived from a common ancestor, they will have similar signatures, which are also called as genetic signatures or the DNA sequences. Genome evolution can be due to different reasons. There are different causes of the genome evolution. Similarity between the different organisms can be due to the sequence composition. In other words, DNA sequence composition. That can be due to the genome organization. Presence or absence of mobile elements which are present on the genomes. Presence or absence of the gene families. So these are some of the points by which a similarity or dissimilarity can be between the genomes or between the individuals. This is genome evolution, chromosome 1. In case of a human, this is chromosome 1 of the humans, of chimpanzee, gorilla, and orangutan. So here if we see that what is the similarity between the chromosome 1 of all these 4 species, we can see that these are the darker bands, and these darker bands, they are almost similar in all the 4 chromosome 1 of 4 species. But if we say here, look here, we can also see here there is a similarity. Here there is another similarity. Same is here towards the centromere, that we can see here the chromosomes, they are similar with each other. And also on the Q arm, here we can see that these 3 are close to each other. And we can also see these 4 bands, they are similar. And also these 4 bands, they are similar. So that genome evolution, as the genomes have been evolved from single ancestors, so there are similarities between the genetic signatures. Here is one of the example in case of chromosome 1 in human, chimpanzee, gorilla, and orangutan. Genome evolution. In any organism's lifetime, genome is used, it is damaged, it is repaired, it is copied and handed down to the offspring cells. In this process, the genome is changed. So these are all the phenomena which are occurring in an organism's lifespan, that the DNA is used, it is damaged, it is repaired, and it is handed down to the offspring cells. What are the basis of change at the genomic level? That the actual basis in the change of the sequence of DNA or the sequence of the genome is mutation, which underlies much of the genome evolution. The earliest forms of the life, they have less number of genes, but after evolution, now the number of genes in the genomes, they are large as compared to the ancestral genes. The size of the genomes have increased over the evolutionary time with extra genetic material, providing raw material for the gene diversification. Initially, genome evolution involved just at a single individual or at a single point. If change have no phenotypic consequences, it determines whether the individual's offspring will carry the mutation or not. This is the phenomenon that how genomes they evolved, how they change with the passage of time. The basic concept is about the mutation or the variants. And if there is a mutation in a genome at a single individual, at a single point, and if that determine, if that is beneficial, then it will go to the next generation. And if it is not beneficial, then it will be eliminated. The process by which frequency of mutations or the frequency of alleles changes in the population is called as genetic drift. These are some of the types of genome evolution, that how genome evolved, types of changes that contribute to genome evolution. Those types, they can be, this is the original genome, and the types of the evolution can be mutation within a gene, that there is a single mutation that can occur in the gene. So this is the genetic innovation to the original genome. The second type can be gene duplication, mean the fragments of the DNA or the genes, they are duplicated. Here there was a single segment. Here we can see that there are two segments. So this phenomena is called as gene duplication. The third can be gene deletion. Some fragments of the DNA or segments of the DNA, they can be deleted, which also works as a fuel for genome evolution. The fourth type can be exon shuffling. So exon shuffling means there are two exons or there are two genes, and the genes, they shuffle on different sister chromatids or between the homologous chromosomes, they shuffle with each other. This phenomena is called as exon shuffling. So there are 4 different types that work as a fuel for genome evolution. Genome evolution can be due to the mutation, it can be due to the gene duplication, it can be due to the gene deletion, and it can be due to the exon shuffling or genome shuffling. Genome evolution can also be due to the horizontal transfer. The phenomena of horizontal transfer is, this is, let's say, organism A and this is organism B. The genes in organism A are shown with different colors, and the genes with organism B is shown with different colors. The horizontal transfer is that in this organism we have seen the same genome or the same DNA or the same genes they have transferred, but in this organism, which was organism B with a new gene, this gene which was marked as red color was actually present in organism A, but here due to horizontal transfer this gene now is with the organism B. So this is called as horizontal transfer, that also works as a fuel for the genome evolution. Mutations that have a beneficial effect, there would be more probability they will be fixed in the next generations. While on the other hand, those genetic variants or those mutations which are detrimental, they have almost no chance of fixation. Substitutions usually refer to as single nucleotide substitutions, but the term indel is also used for the substitutions. So if we conclude that what is genome evolution, during the lifetime of any organism, DNA is used, DNA is copied, it is handed over to the next generation, mean to the daughter cells. So during all this process, DNA is changed.